Hi there, today's chapter would be photosynthesis. Do feel free to correct me if there's any inaccuracy or if there's a need to clarify. We would start with what we have learned previously in biology. In AS, we learned that chloroplast is around 3 to 10 micrometers in diameter, and it also has 2 membranes and 70S ribosomes and a circular DNA. In A2, we learned that stroma is the site of light-independent stage, also known as the Calvin cycle. It also contains enzymes like rubisco and also sugars, lipids, starch, ribosomes, and DNA. At the internal membrane system, we have the thylakoid membrane, which is the site of light-dependent stage. The thylakoids occur in stacks called grana. Grana hold photosynthetic pigments and have a large surface area for maximum light absorption. The pigments are arranged in light-harvesting clusters, or we can also call it the photosystems. The primary pigment is surrounded by accessory pigments. The role of accessory pigments is to absorb light energy and to pass the energy to primary pigment. Different photosystems absorb light at different wavelengths to maximize light absorbed for photosynthesis. The membranes also hold ATP synthase, or you can also say electron carriers, for photophosphorylation. So if there is a question asking to describe about the role of chloroplast pigments, you can say that pigments are arranged in light-harvesting clusters, forming photosystems on thylakoid membranes. The accessory pigments surround primary pigment. Examples of accessory pigments are chlorophyll B, carotene, and xanthophyll, while an example of primary pigment is chlorophyll A. Accessory pigment absorb light energy to excite electrons and passes the energy to primary pigment. The pigments absorb light energy at different wavelengths to maximize the light energy absorbed for photosynthesis. Next, we will talk about the light-dependent and light-independent stage. During photosynthesis, light-dependent stage, which occurs at the thylakoid membrane, would occur first, followed by the light-independent stage at the stroma. During light-dependent stage, cyclic and non-cyclic photophosphorylation occurs. Do take note that it is photophosphorylation and not phosphorylation, which you learned previously in energy and respiration. During light-dependent stage, light energy will be absorbed by the chlorophyll A, and this would result in electrons to be excited. The electrons would then be emitted from chlorophyll A, resulting to the photolysis of water. Hydrogen ions would be released from photosystem 2, while the electrons would be released from photosystem 1. The released hydrogen ions and electrons would combine with NADP to form reduced NADP. The energetic electrons would release energy as they pass through the electron transport chain. The released energy is used to transfer protons across the thylakoid membrane. The protons will return to the stroma from the thylakoid space by vacillated diffusion through ATP synthase, providing energy for ATP synthesis. If the question asks to explain photophosphorylation, you could explain using these 3 last points. As I said earlier, cyclic and non-cyclic photophosphorylation occurs during the light-dependent stage. During cyclic photophosphorylation, only photosystem 1 is involved. Photoactivation of chlorophyll occurs and ATP is synthesized in the end. While during non-cyclic photophosphorylation, both photosystem 1 and photosystem 2 are involved. Photoactivation of chlorophyll occurs likewise, and there is also an oxygen evolving complex which catalyzes the photolysis of water. ATP and reduced NADP are synthesized in the end. After the light-dependent stage, we would have the light-independent stage, also known as the Calvin cycle. There are 3 main stages of the Calvin cycle. First off, the rubisco will catalyze the fixation of carbon dioxide by combination with a molecule of ribulose biphosphate, which is a 5-carbon compound, to yield 2 molecules of glycerate 3-phosphate, which is a 3-carbon compound. Then, the glycerate 3-phosphate is reduced to triose phosphate in reactions involving reduced NADP and ATP from light-dependent stage. Lastly, the ribulose biphosphate will be regenerated from triose phosphate in reactions that use ATP. The intermediates of Calvin cycle is used to produce other molecules. For example, glycerate 3-phosphate would produce some amino acids, while triose phosphate would produce carbohydrates, lipids, and amino acids. Moving on, The absorption spectrum is a graph of the absorbance of different wavelengths of light by a pigment, while action spectrum is a graph of the rate of photosynthesis at different wavelengths of light. Do remember that short wavelengths mean more energy, as you may be asked to interpret the graphs. You can use chromatography to identify and separate pigments. To separate pigments, you should first grind the leaf and use solvent to produce a leaf extract in which contains mixture of pigments. Draw a pencil line around 2 cm above the base and place the paper vertically in jar of solvent. The solvent would then rises up the paper. Each pigment will travel at different speed, hence the pigments will be separated as they ascend. The distance moved by each pigment is unique, and it can be used to calculate RF value using the formula distance moved by spot divided by distance moved by solvent. Two-dimensional chromatography would produce better separation of pigments. To identify pigments, you should place a spot on the baseline and concentrate the spot. Place the base of chromatogram in solvent and cover to stop the evaporation of solvent. Calculate RF value by using the formula distance moved by spot divided by distance moved by solvent. To identify the pigment, compare the calculated RF value with known RF value. To explain limiting factor, you can say that photosynthesis is controlled by several factors. Limiting factor is the factor in shorter supply which prevents increase in rate. Examples of limiting factors are light intensity, carbon dioxide concentration, and temperature. An increase of light intensity, carbon dioxide concentration, and temperature will lead to the increase of rate of photosynthesis. However, from the graph example on the right side, you can see that although the light intensity is still increasing, the rate of photosynthesis remains constant. This is because other factors like temperature or carbon dioxide concentration could be the limiting factor preventing the increase in rate of photosynthesis. To investigate the effect of wavelength of light on the rate of photosynthesis of a plant using redox indicators such as DCPIP, you can use Hill reaction. Oxidized DCPIP is blue, and it would go colorless when it is reduced. First, you would have to crush the leaves in acetone to make chloroplast extract. Then, expose the chloroplast and DCPIP to wavelength of light and measure the time taken for blue to disappear. You can calculate the rate of photosynthesis using the formula 1 divided by time taken for blue to disappear. Test 5 or more wavelengths and do 3 repeats. To plot a graph, plot wavelength on x-axis against calculated rate on y-axis. To investigate the effect of temperature on the rate of photosynthesis of an aquatic plant, you should use a microburette. First, cut the shoot of aquatic plant and place the shoot in tube of hydrogen carbonate solution to provide carbon dioxide. Choose 4 different temperatures to carry out the experiment. For example, 20, 25, 30, and 35 degrees Celsius. Use a water bath to maintain temperature and allow plant acclimatization so that the plant can adapt. Place the lamp at a fixed distance away and use a heat shield. You can obtain the results by counting the number of bubbles produced in set time. Repeat the experiment twice more and calculate mean values. That would be all for this chapter. You can find the note for this chapter in the description box below. Thank you.